Oh, welcome everybody to our today's seminar on Do Nutrients Limit Soil Carbon Sequestration? For those of you who haven't been before, these are sponsored by AGU and the Tri-Society, so the Soil Science Society of America, the American Society of Agronomy, and the Crop Science Society of America. And we've been doing these about once a month. And for the next few months, the schedule is going to get a little different. So if you can just keep posted to the announcements from the societies, and you can always see where these seminars are hosted on the International Soil Carbon Network website, ISCN. You can also find the old seminars there if you've missed some of them, and this one will be recorded and placed there as well. So we'll just give it about one more minute to let people filter in, and then we'll start with the presentations. Okay, so I'll introduce our two speakers, and they'll each talk for about 20 minutes, and then we'll have about 8 to 10 minutes for questions for each of them. And while you're listening, if you just want to add any questions you have you can ask them on the questions. So we'll address some of those as well as some of the questions that people put on their registration forms. So our first speaker is going to be Adrian Keller. Oh, I guess I should introduce myself too, sorry. So I'm Dan Lipson. I'm a research soil scientist at the Soil Health Institute. I'm one of the co-organizers of this seminar series. And I'm pleased to introduce our two speakers today. The first one is Adrian Keller, who's a postdoctoral research scientist in Dr. Sarah Hobby's lab at the University of Minnesota. She's working with the Nutrient Network, a coordinated research network that includes more than 130 grassland sites worldwide to examine the effects of nutrient pollution and altered grazing regimes on grassland, plant, and soil properties. Adrian is also a researcher with the new Minneapolis-St. Paul metropolitan area urban long-term ecological research site, where she is working on a collaborative interdisciplinary project examining how social and ecological factors influence canopy diversity and resilience across the Minneapolis-St. Paul metro area. And our second speaker, Stuart Grandy, is a professor of soil fertility and biogeochemistry and co-director of the UNH Center of Soil Biogeochemistry and Microbial Ecology. His lab examines how agroecosystem management influences soil health, as well as the microbial processes driving soil organic matter formation and nutrient cycling. The lab is currently focused on the processes regulating from where and how plants and microbes acquire bioavailable nitrogen. And so with that, we'll start with Adrian's talk on increased nutrient supply does not consistently increase soil carbon storage across 9 natural grassland sites.
Great, thanks, Dan. It's great to be here. Thanks for everyone for joining. And I'm really excited to talk about some of the work that I've been working on with the Nutrient Network. And I'm going to be focusing on 9 sites within the temperate grasslands in the US. So I'm going to focus my talk on natural grassland systems and nutrient effects in those systems. And then I think it'll be a really interesting comparison and ability to contrast with agricultural systems that Stuart's going to be talking about. And so this work has been done with support from the National Science Foundation and with a whole host of fantastic collaborators. All right, now we have the challenge of advancing. All right. So I thought we would start by setting the scene and looking at global patterns of soil organic carbon. And so this is a map that is showing the distribution of soil carbon that's stored across these different systems. And so you can see there's a wide variety of variation in how much carbon is stored across these different systems. I'm going to be focusing in on the temperate systems in the United States, and particularly in grasslands. And so grasslands make up approximately a third of non-agricultural land across the globe, and they store about 20% of our soil carbon. So they're a really important system to be thinking about as we think about climate mitigation strategies and soil carbon storage. And so here's a diagram that maybe many of you have seen before that's showing the distribution of carbon in terrestrial systems, disregarding permafrost. So we see, if we don't account for permafrost, we see that more than 50% of our carbon is stored in the soils. And so I think this really speaks to the potential for soils to serve as a natural climate solution. And so there's a lot of focus on how can we get more carbon in the soil and to take it out of the atmosphere to mitigate climate change. And so that's done right by increasing vegetation biomass, and then a lot of that biomass is going to get sequestered into the soil. Some of it is going to get respired back into the atmosphere initially, and some of the vegetation that ends up in the soil can get sequestered away for long periods of time, and other inputs will be fairly quickly recycled back into the atmosphere. And so today we're really thinking about how, if we add nutrients, how might this increase inputs from the system in terms of vegetation, and how might that alter outputs from the soil. And so it's this balance between inputs and outputs that's really going to dictate the potential to sequester carbon over a long period of time. And so I'm going to sort of flip a traditional presentation style today, and I'm going to present both my research questions and the take-homes all at once, and then we'll kind of walk through those. So I think the question many of you came here today to learn about is, if we add nutrients to natural grasslands, can we reliably increase soil carbon? And so the answer that I'm going to show is no. And so this begs the question, why not? And this could be a detectability issue. So it could be that we're trying to detect small changes in soil carbon within a really big soil carbon pool, right? Because we have, as we showed at the very beginning, these pools of carbon are quite large. It could also be a detectability over time, and that we just haven't experimentally run out these experiments to be able to detect changes at the time scale that's relevant to nutrient effects on soil carbon. So that could be part of the story, and I'll speak to that a little bit. It could also be that perhaps across sites, there's just minimal potential to increase soil carbon. But I'm going to show that at least at some sites, that's not the case, because we do see increases in soil carbon at some sites, but not at others. So there seems to be this site context dependency, as it's common across ecological studies, and that sites are responding differently to nutrient addition in terms of soil carbon dynamics. And so then I thought, let's see if we can identify different site factors that might help us predict where nutrient addition can have the largest effect on soil carbon sequestration. So this is an interesting question just as a basic ecologist, but I think it also is really important if we're trying to think about how do we best manage our lands to help mitigate climate change and store more carbon for long periods of time below ground. And so we don't quite have the answer yet, but I think I'll show some compelling and interesting data that speak to a couple of different site factors, in particular soil mineralogy and climate or moisture index, that seem to be important. And so I think that there's some follow-up questions from this study to try and understand this site context dependency a little bit better. And so I'd like to end with what's hopefully will be a lively discussion and questions and future research needs. Okay, so I just really briefly want to introduce the Nutrient Network for those of you that may not be familiar with it. So the Nutrient Network is a distributed and coordinated research network with sites across the globe. And at each of these sites, the same experimental design is replicated in a fully factorial block design. And so in each plot or each block, there's a nitrogen addition, a phosphorus addition, potassium and micronutrients, and each of those in full factorial. And so this is replicated across many sites, and I'm going to be talking about just 9 sites today. And so these are the 9 sites. We can see that they're distributed across the United States, and they vary in ecosystem type. So out in Oregon, the Lookout and Bunchgrass sites are these wet montane grasslands. Shortgrass steppes, so the SGS, sort of in the middle of the country, is shortgrass prairie. And then, for example, Trelease out in Illinois towards the east is a tall grass prairie. So we have a variety of different types of grasslands that we're capturing in this study. And we also have differences in the amount of atmospheric nitrogen deposition or external nutrient supply, regardless of the fertilization treatment that these sites are experiencing. And importantly, this nitrogen deposition metric is also decoupled from climate. And so we have a big range in climate and moisture availability that these sites are experiencing, and that's distinct from the atmospheric nitrogen deposition that they're also experiencing. So we have a wide diversity of sites to explore the question of how nutrients are influencing soil carbon. So as I already alluded to, we first wanted to look at across sites, if we add nitrogen yearly for a whole decade, do we see changes in soil carbon? And we expected that we might see increases in inputs, and then that should translate to potentially to increases in soil carbon. But yet across these 9 sites, we don't see any significant differences between treatments in terms of total soil carbon pools. So this is perhaps a bit surprising. But the next thing we wanted to do is that we thought, again, maybe this is a detectability issue. So maybe it's just that we can't detect relatively small changes in soil carbon in this huge pool of soil. And so then we wanted to look at distinct soil carbon fractions that are operationally defined, but tend to relate to the properties of those soil fractions and the rate at which they persist in the soil. And so we split up into 3 different fractions. First, the light particulate organic matter fraction, and this is made up primarily of those fresh inputs from vegetation, and it's relatively fast cycling. And so this fraction has relatively low potential to really increase long-term carbon sequestration. And the second fraction we looked at was the heavy particulate organic matter fraction, and this tends to be made up of macro aggregates, and it can be heavily influenced by particularly below ground plant inputs, root inputs. And then finally, the fraction that is of particular interest to long-term soil carbon sequestration, the mineral associated organic matter fraction, and this tends to be the most slow cycling soil pool, and therefore has high carbon sequestration potential. So there's a lot of emphasis on how do we increase this mineral associated organic matter fraction. And so then we looked again across these sites if there was any evidence for nutrient effects on these 3 different fractions. And so we don't see strong effects in the light particular organic matter fraction, although perhaps a slight trend in increasing that pool with nutrient addition. No significant change in the macro aggregates, and similarly, no statistically significant effect of nutrients on this MAM or mineral associated organic matter fraction. There is a slight trend towards increasing the slow cycling MAM pool with nutrient addition, and so this could hint at that we just haven't had enough time to detect changes, and perhaps with several more decades of experimental fertilization, we might see this mineral associated organic matter pool grow in these fertilized treatments. And so perhaps this is both hints at a detectability issue, both in pool size and in duration of the experiment. Okay, so why do we not see consistent fertilization effects on soil carbon pools? So this is the same figure that I showed initially, but now I've mapped on the site-specific responses. So each of these points is the mean for a given treatment at a given site. And so you can see that while there is not a lot of variation across treatments in terms of soil carbon pools, that we do see this huge variation in soil carbon pool within any given treatment. And so this really hints at the fact that there might be some really important site factors that are driving these soil carbon dynamics. And so you might be wondering what these two, the light blue and the gray points are. So those are the wet montane sites in Oregon, Lookout and Bunchgrass. And you can keep your eye on those because they tend to sort of pop out in some of the data analyses. And similarly, we are looking at the data in the same way with these different fractions. And again, across the board, we see really strong differences within a treatment across sites. And that very much overwhelms any fertilization effect across the sites. And so then we thought, okay, well, our hypothesis was you increase nutrients, that should increase inputs. Do we see that? So here I'm showing the log response ratio, or the relative increase in above ground biomass in our fertilized versus the control plots. So here we see all these values are above 0. So if you add nitrogen, phosphorus, or nitrogen and phosphorus in combination, we do see increases in above ground biomass compared to the control. But we don't see a similar response below ground, and it's much more variable. So it looks like while above ground inputs are increasing with nutrient addition, we don't see significant increases in below ground biomass. And in fact, we would expect that there would be a relationship between between vegetation responses and soil carbon responses. And so we might expect on this plot where we're plotting nutrient effects on above ground biomass and nutrient effects on soil carbon stocks. We predicted that there would be sort of a positive relationship between these two axes, and yet we really see no relationship. So this is further evidence that the effects we expected to see on nutrient inputs or effects we expected to see on vegetation were much weaker below ground and really did not translate into any significant effects on soil carbon stocks. Okay, so then we want to see if we could identify site factors where the nutrient addition might have the largest effect on soil carbon sequestration. So we didn't see the strong direct relationship between adding nutrients and increasing soil carbon stocks. So then we thought perhaps there are factors of site fertility that would interact with these nutrient additions to influence soil carbon. But across the board, we didn't see a lot of evidence that site fertility interacted with nutrient effects. There was a weak nitrogen deposition by phosphorus addition interaction, but that in itself didn't explain the soil carbon responses that we were seeing across these different sites. In contrast, we saw a couple of factors, soil factors that didn't interact with the nutrient treatments, but were important in predicting soil carbon patterns. So soil texture, and particularly soil aluminum and iron, or the soil mineralogy, was really important in predicting these soil carbon stocks. And so we put these factors into, for those of you that are familiar with statistics, into a linear mixed model, and we found that these fixed effects, or these site effects in addition to the nutrient treatments, explained about 6% of the variation in soil organic carbon. And that left about 69% of variation in soil organic carbon that could be, that was explained by these unmeasured site factors. So that site being used as a random effect in the model. And then there's our remaining 25% that was completely unexplained by our statistical model. However, at these sites, the soil mineralogy, or aluminum and iron, was highly correlated with moisture index. So moisture index is a climate variable, so it's mean annual precipitation divided by potential evapotranspiration. So they are highly correlated in that these Oregon sites in particular had really high aluminum and iron oxides, as well as a really high moisture index for its climate. So we couldn't tease this apart statistically, so we ran a separate model that included just the moisture index. And here we saw that these site factors explained a lot of variation in soil carbon, leaving a little variation unexplained as the random site effect. And again, a similar, about a quarter of the total variation was unexplained by our model. So those were the statistical results. And now I'm going to actually show you the data that went into it. And the take home is really that these mineralogy, in particular, these poorly crystalline amorphous iron oxides, so there's aluminum and iron content, and or climate, but not nutrient supply, seem to explain these soil carbon pools. So here we're seeing on the x-axis the oxalate extractable aluminum and iron. So that was the soil mineralogy factor that we looked at before. And we see a really strong positive relationship between soil mineralogy and soil carbon. And this is regardless of fertilization treatment, so across all the plots. And we see a similar pattern with moisture index. And here, if we look at just percent silt clay, so the soil texture, which is a common factor that is included in a lot of ecosystem models that are trying to predict carbon dynamics across different sites, we see that in general, there's a positive relationship between soil texture or percent silt clay and soil carbon. But these two Oregon wet montane sites have really high aluminum and iron, but they sort of fall out on this relationship. And so then we wanted to look at these same patterns. So the above row, the first row is what we just looked at, and then the second row is looking at just the slow cycling pool with mineral-associated organic matter. And so we again see that there's something interesting going on with this mineralogy, where the sites that have relatively low clay, but a really high oxalate extractable, these aluminum and iron oxides, really don't follow these relationships that are seen at the rest of the sites. And so I think this is an important next step, is thinking about the role of soil mineralogy in driving soil carbon stabilization, and how important factors such as soil mineralogy may be in incorporating into ecosystem models. And are these two sites more or less anomalies, or do we need to be incorporating some metric such as iron and aluminum and oxides, aluminum and iron oxides into our ecosystem models to really predict where carbon is going to be, where carbon storage is going to be highest across our different ecosystems. Okay, so we've seen that we can't reliably increase soil carbon across grasslands if we add nutrients, and this might be in part due to a detectability issue, but it seems like there is some interesting site-specific factors, such as mineralogy and climate, that could be driving these patterns. And so I thought I would just leave us with some sort of open questions and future research needs. So I think there's increasing evidence that below ground inputs are particularly important in driving both soil carbon formation and the long-term persistence of this carbon in our soils. And we still have a pretty poor idea of how below ground inputs regulate these soil carbon dynamics. And so we see some evidence that in the absence of roots, that if you add nutrients, decomposition decreases. And so you actually might add nutrients and suppress decomposition, and that should then promote more soil carbon storage. And so this is some preliminary data from Lang Delancey showing that soil respiration, which is a proxy of decomposition, is suppressed in these nutrient treatment effects. And this is across the same grasslands that I've been presenting on. But what happens when we add roots? So this is in little jars without roots, and I think there's an important role of both root inputs and then active priming of the organic matter, and really understanding these dynamics is going to be important for thinking about nutrient effects on long-term soil carbon storage. And then I think an underexplored arena of this whole question of soil carbon storage is the role of the organic horizon. And so the organic horizon, which grasslands don't have a strong organic horizon, but the organic horizon has this unique physiochemical properties and also has unique decomposer communities. And so you tend to see stronger nutrient effects in forests where you do often have a fairly significant organic horizon. And so perhaps this contrast between forests and grasslands may be due in part to simply the presence or absence of the organic horizon. And so this is shown here in some work by Sarita Frey and others, showing that especially at the lower ends of nitrogen addition, the increase in soil carbon stocks are really just observed in this organic horizon, or that lighter color bar. And then I think, of course, we're living not just in a world where we're seeing increased nutrient supply, but we're also seeing a whole host of other global changes, such as changes in temperature and precipitation that then can shift vegetation and resource availability. And so thinking about how nutrient supply is interacting with these other global changes is going to be important for accurately predicting soil carbon dynamics. And then finally, I'll just end with sort of a plug, and I think that it will be becoming increasingly important to integrate different disciplines of soil science. And so I think traditionally there's been soil ecologists and so that think about the biology of the soil, and that they might even be housed in different academic departments thinking about soils in very different ways than those thinking more about the soil physics and the soil chemical properties. I think integrating these two disciplines can be really useful for trying to understand soil carbon dynamics in the changing world. And so I will leave it at that, and I think there's maybe time for a few questions. So thank you very much.
Well, thanks, Adrian. That was a great presentation. And so you can, I think we have plenty of questions there, but if you have more, you can keep adding them. But the first question that came up a couple times, there were people interested in what depth these samples were.
Sure, yeah, that's a great question. So we took samples from 0 to 15 centimeters and 15 to 30 centimeters, and we really didn't see strong depth differences at those two depths, but I think there could be stronger depth differences if we went all the way down to a meter. But I think a lot of the majority of the root biomass is going to be in the first 30 centimeters, and some of these sites actually don't have a rooting depth much below that 30 centimeters. So I think we captured a lot of the more dynamic plant microbe soil interactions, but we would see very different responses, I would expect, at depth, where you're not seeing as much of that like active root and rhizosphere soil.
Okay, great. And there's some questions, you mentioned what you found to be significant predictors, but for people who aren't familiar with the data set, there are some questions about, other things that you measured that didn't turn out to be significant. And so could you comment, like some of the things that came up were like pH or inorganic nutrient concentrations or microbial biomass? I assume there's like a long list of things that
were measured. That's awesome. Yeah, so right, so we didn't see any, factors such as basically everything you just explained. So baseline soil carbon, so soil carbon in control plots wasn't predictive of how those sites would respond to fertilization. Baseline fertility, so total nitrogen and inorganic nitrogen wasn't predictive. Interestingly, when we added nutrients, you tend to see a soil acidification or a reduction in soil pH, and we didn't see strong reductions in pH. There's some interesting work at some of these sites that suggests that acidification effect can take a little while to manifest, and I would have expected it to have manifested after 10 years of fertilization, but perhaps that's yet to come. And so we didn't see a strong pH effect, and therefore we didn't see any relationship between pH and these nutrient effects. And we also didn't see a strong, so we saw this moisture index effect, but other factors of climate, such as temperature, didn't seem to be as important.
Okay, and then there were some questions about, so the Oregon sites that were very different in terms of mineralogy. There was a question about whether those were andisols. Do you know?
Yeah, so these are really young volcanic soils, and so they look. So we had a variety of different soil types across these 9 sites, and so if you going sort of connecting to your previous question, if you just put in ecosystem type or soil type, that wasn't predictive of the response. But it seems like there's something really particular to these young, underdeveloped volcanic soils that have low clay content, but these high reactive minerals that seem to be particularly important in predicting soil carbon. So it's not so much the ecosystem type or like the plant community or sort of the broad soil order, except for with these organ sites, if that makes sense.
Okay, and then do you have, so you have a wide range of sites that are in here, but the network is much bigger than that. If you start thinking even more broadly than the sites that you've presented today, would you imagine that you would see, if you added a bunch more kind of high aluminum and iron sites, but in a different climate or in tropical soils, or do you think these patterns would hold as you expanded this even to more sites?
Yeah, and I think there's some other compelling research that suggests that this context of soil mineralogy beyond soil texture is really predictive of soil carbon storage, especially these persistent soil carbon storage. And so the short answer is I would expect there to be this relationship to hold across broader sites due to the mechanistic physicochemical processes that are driving these relationships. And I think there is interest in the nutrient network to try to see if we can get a broader range, more data that spans a broad range of soil mineralogy. And so I think that's sort of in the brainstorms in terms of next steps with the nutrient network. And I think one of the things with just doing 9 sites is that you're going to find a little bit of a gradient, but then something is always going to be a little bit of an outlier. But I would expect this relationship to hold across sites if we were to expand it to the whole nutrient network.
Great. Well, thanks, Adrian. Now we're going to switch over to our second speaker, Stuart Grandy. Great. Well,
Dan, what can you see?
It just looks black right now. Do you want to give it back to me and I'll...
Yeah. Let's see. Okay. Do you have it back?
Yep. So let me give it back to you.
Now, do you see the first slide?
No, it's just black again.
Huh. Okay.
Should try one more.
Let's try one more time.
Yeah.
So we were having trouble at times with this earlier. Let's see. All right. Let me change my presentation around. Okay, so if that makes any difference. Okay, go ahead and try again, Dan.
Okay.
I'm getting this note that the screen sharing is paused because there's another window on top of your shared application.
Oh,
Can you see that?
Yep.
Oh, good. Okay. No.
Oh, it was there for a minute and now it left again. Oh, there we go.
Okay, I think it's okay. And when I transition it, you can see it?
Yep.
Great. All right.
Okay. Thanks,
Thanks
Stuart.
everyone for your patience. It's great to be here, and I really appreciate the invitation from AGU and the other organizers of this event. And thanks, Adrian, for your great talk and setting up my talk, which is really not about grasslands. Well, there are converted grasslands. They're more about agricultural sites and trying to think about how we build soil carbon in agricultural sites and sort of flipping the question on its head and thinking not about carbon per se, but how do we build nitrogen in these systems where nitrogen losses are very, very high, and we tend to export a lot of nitrogen, and the crop residue carbon to nitrogen ratios are very, very high, much higher than the soil organic matter carbon to nitrogen ratios. We face this dilemma: how do we get enough nitrogen into these agricultural soils to build soil carbon? There's no doubt that we have opportunities to build soil carbon in agroecosystems. So here is a prairie. We did some back of the envelope calculations and come up with a carbon content in this prairie soil of about 165,000 kilograms of carbon per hectare to 30 centimeters using a bulk density just over 1. That site converted to an agricultural site might have a carbon content of about 49,500 kilograms of carbon per hectare using similar assumptions. That's a massive loss of carbon, and that deficit represents a potential for carbon sequestration. But at the same time we've lost carbon, we've also lost nitrogen. Our prairies, when we moved west, were extraordinarily productive without any fertilizer inputs. And that's because of the massive amounts of nitrogen that they stored. This prairie might have close to 14,000 kilograms of nitrogen per hectare per year. And if you think about generating a bumper corn crop at 150 kilograms of N per hectare, 200, something like that with modern varieties, you can see that we had a lot of years of productivity built up in those soils. In contrast, agricultural soils may have about 4,000 kilograms per hectare. So at the same time that we're thinking about building up carbon, we also have to be thinking about how are we going to be building up that nitrogen in the soil? How do we get enough nitrogen into organic forms in these agroecosystems to sequester carbon? So how do we increase organic matter nitrogen? Let's just use the 4 per mil initiative for a minute. If you look globally, that would be an increase of about 1,200 teragrams of carbon per year and 100 teragrams of nitrogen per year, assuming a soil C to N ratio of 12 to 1. To get this much nitrogen into the soil would be an increase of about 75% of current global nitrogen fertilizer production, or doubling symbiotic nitrogen fixation. And on a per hectare basis, it represents, because I find it helpful sometimes to think about things on a per hectare basis because I come from somewhat of a management background. About 17 to 50 kilograms of nitrogen per hectare per year have to be stored in soils in an organic form to build up enough carbon to meet this 4 per mil initiative. At the same time, we already have a very inefficient nitrogen system in agriculture. This inefficiency harms drinking water, it stimulates algal blooms in dead zones, it accelerates N2O production. How much fertilizer nitrogen is recovered in crops? It varies depending on the region, depending on the crop, 40% or so. That means that 60% of the nitrogen is not being taken up by the crop that's applied in any given year via external inorganic, typically inputs. And at the same time, it also suggests, and we know, that a whole lot of nitrogen that plants take up in any given year originated with the organic matter. And I also tend to think that a lot of fertilizer that we add cycles through organic matter quite quickly, although that's not been shown to date across a wide range of studies. So we have to build more nitrogen in soils if we're going to sequester carbon, but we already have this tremendous problem of nitrogen inefficiency in agroecosystems. And so what I want to talk about today is some of the work that we're doing, the experimental work, the conceptual work. I'm not going to get so much into our modeling work today related to nitrogen, where it's stored, how plants get access to it, and throw out some ideas for increasing nitrogen use efficiency. And I'd like to point out that this problem of nitrogen use efficiency is only going to be accelerated with a changing climate. In particular, the extreme dry to wet transitions that we expect across much of the Midwest are already increasing nitrogen loss in some years. And what happens is that nitrogen accumulates via mineralization as soils dry, because microbes can continue mineralizing when soils are dry, whereas plants can't really take up that nitrogen. And so you get this accumulation of inorganic N, and then we get these massive rainfalls, and boom, it creates a whiplash effect, and lots of nitrogen is lost from the soil, leaches out, denitrifies, and lowers our nitrogen use efficiency and contributes to diminishing environmental quality. So how do we tighten the nitrogen cycle in agroecosystems? Those of you familiar with agricultural management practices know that the most common approach is the 4 Rs, which provide very modest NUE improvement. And those involve basically fertilizer engineering approaches where we manipulate the rate, time, location, and form of nitrogen fertilizer. You can imagine banding, or split applications, or changing the form of nitrogen. At the same time that these have provided only modest increases in NUE, we've learned some, but not enough, about in situ bioavailable nitrogen cycling related to organic matter. And I would argue that we have focused too little within the nitrogen cycle on these fundamental processes of bioavailable N cycling and organic matter nitrogen turnover, in part because they're very, very complex and controlled by a lot of different biogeochemical processes and enzyme system within microbes. So what originally got me interested in this work was when I was a graduate student at the Kellogg Biological Station, and I would look at these prairie soils at KBS. And not every prairie is the same, and I know that some are different than this, but the nitrogen cycling in those soils was extraordinarily tight. Very little N2O loss, very little nitrogen leaching. Inorganic N was often unmeasurable, and yet they maintained extremely high productivity. And it just got me wondering about the plant-soil microbe interactions that are generating enough nitrogen to sustain the productivity in these sorts of prairie soils. And from this, we developed a new framework for beginning to do this work. This was originally conceived by Andrea Yilling, and then Amanda Daly has followed up with it recently, where we take the original view of Josh Schimmels and colleagues focused on particulate organic matter conversion to dissolved organic N and then mineralization, and really incorporate the role of the soil interface, in particular the mineral associated organic matter and the microbes and the plants. And so we move this system to one where there are potentially 3 major sinks for nitrogen in soils: the mineral associated organic matter, the plants, and the microbes. And the many controls over these and their interactions create a much more complex biogeochemical system driving the nitrogen cycle. And that's what we've been trying to understand, with a particular focus of late on the mineral associated organic matter pool, which may be an overlooked but very large source of nitrogen. This pool in many agricultural soils contains 85% or so of the total nitrogen stored in organic matter. And also, if you look at the chemical properties of MAM, it's more favorable to microbial and plant access, whereas POM has a high C/N ratio, consists of partially decomposed litter fragments, and is often associated with nitrogen immobilization in the lab. MAM has a very low C/N ratio. It consists of smaller nitrogen-rich molecules such as proteins, amino sugars, and in the laboratory, it's often associated with nitrogen mineralization. So how do we build MAM-N? What is the role of the soil community? And then I'm going to come around to how would plants and microbes actually access this pool of nitrogen that's historically been thought to be relatively inert. So I'm going to go back to some of the work that Cynthia Kallenbach did and show that the microbial traits seem to really matter for building up MAM. She was able to show in this low crop complexity and high crop complexity site at the Kellogg Biological Station that the carbon use efficiency of the microbial community was very closely related to the soil organic carbon, and that we saw much higher CUE under high crop complexity. This translated to higher soil organic carbon. And we in particular saw that that soil organic carbon was made up of microbial lipids and nitrogen bearing compounds that were in the MAM pool. And so this suggests that the microbial traits really matter for building MAM carbon and also MAM nitrogen. Similarly, in terms of scaling it back out to management, Maria Moshammer saw something very similar in a paper that we have in press with Tim Bowles at Soil Biology and Biochemistry, where we saw that higher crop rotation at a different site in Mead, Nebraska changed the chemical diversity of inputs. We saw a shift in microbial trait changes, including variation in enzyme expression, creating a positive feedback on soil organic matter. And in particular, we saw more soil organic carbon in MAM, possessing a strong microbial signature under higher temporal diversity. So again, we're seeing where we incorporate greater temporal diversity into our cropping systems. We're building up both MAM carbon and nitrogen that appears to be associated with shifts in the microbial community. So how do plants access MAM, and what is the role of plant microbe interactions in this? And I'm going to come back to Andrea Yilling's conceptual model, where she really nicely laid out 2 different mechanisms by which rhizosphere interactions can generate MAM nutrients. And in one, oxalic acid mobilizes metals, displaces MAM, and may stimulate oxidase production. And I'm going to show that we've actually now seen this in laboratory experiments. And also glucose can stimulate microbial enzyme production and microbial activation and nitrogen mining response of MAM nitrogen. And in a follow-up laboratory experiment that Andrea published last year, we saw that Oxidative enzyme activities were increased by oxalic acid additions to soils, whereas hydrolase enzymes, both CBH or cell biohydrolase and beta glucosidase, were increased by glucose additions. Interestingly, we also saw that oxalic acid increased metals. Both iron and aluminum were mobilized by the presence of oxidative enzymes, suggesting, consistent with their pathway, that oxidative enzymes and metals are being mobilized by oxalic acid, and these hydrolase enzymes are being mobilized or activated and increasing due to glucose additions. And associated with that, we did see changes in nitrogen in the soil solution using isotopes. So we think that the MEAM pool, this is from Amanda Daly's paper last year, provides a place to accumulate, retain, and recycle massive amounts of soil N. And to optimize this pool and to encourage sustainable productivity, we should be thinking about how to build this pool, provide microbes and plants access to it, and to augment it strategically with external fertilization. And to show now a study with plants, I'm coming to a paper that Tim Bowles published in 2022. It's just, I think, come out recently in Soil Biology and Biochemistry, or at least the preprints available there now. And we looked at different crop rotations. Again, this was at KBS. We looked at corn, corn, soy, corn and a legume cover crop, and corn, soy, wheat with a rye clover cover crop. So if you look across this crop complexity gradient, we're seeing an increase in both carbon and nitrogen. Look at where that increase is happening. It's happening in this really, really fine fraction, less than 20 microns. It was not happening in the coarse silt and sand fractions. We're seeing these increases in MEAM nitrogen. So how did that affect plant growth? We grew plants in the greenhouse in these different soils across this crop complexity gradient. And we looked at plant nitrogen concentrations. You can see they were much, much greater where we had more MEAM N, much higher plant nitrogen concentrations, and this was without fertilization. We also looked at the proportion of nitrogen from new 15N labeled clover that we added to these soils in the different treatments, and the simple rotations and the non-rotated corn were much more dependent on the new inputs from clover, whereas the corn, soy, wheat rotation was depending on the in situ nitrogen, and much of that was coming from MEAM nitrogen, we believe, because that's where the increase occurred. We also saw changes in photosynthetic rates. Overall, we saw the complex crop rotations increased MEAM N and microbial community size, increased the uptake of nitrogen from soil organic matter nitrogen pools, reduced nitrogen uptake from new nitrogen inputs, and increased photosynthetic rates, stomatal conductance, and interestingly, intrinsic water use efficiency. So again, I think this paper is showing a really nice direct link between an increase in this MEAM N pool thanks to cropping system complexity and the potential crop productivity generated from in situ nitrogen cycling. So as we move forward, I think that we have to think about nitrogen and how it cycles differently and what we might consider low soil health and high soil health systems. Low soil health systems are fertilizer intensive. They are going to depend entirely on external inputs. In a high soil health system, we have the potential for building up nitrogen pools in different places in the soil, in different forms, and activating plant microbe mineral interactions to access these pools. So on the right-hand side, where we show this much more complicated high soil health system, you can see these different sorts of interactions that we might expect would generate plant nitrogen. With bubble A, we have the production of plant growth promoting hormones by microbes that are feeding on root exudates. This is a mechanism that's just recently been further clarified in the literature. In B, we're seeing, consistent with Andrea Yilling's work, the production of reaction oxygen species and enzymes going after MAOM-N. In C, we have the classic microbial loop where we have protozoans and nematodes gobbling up bacteria and releasing nitrogen through sloppy eating. And then we bring in one of my favorite structural units, the aggregates, where we have in D, the plant root is growing through an aggregate in these healthy soils, and it splits it open and oxygen comes in and nitrogen becomes mineralized. And then in the other one, we have in E, we have mycorrhizal hyphae growing into these aggregates and accessing occluded N. So in these healthy soils, we have all of these pathways by which we can be generating N, many of them involving MAOM and the buildup of MAOM nitrogen in these soils. I also want to point out, and it's not the topic of this talk, but these very different nitrogen systems provide a real challenge for soil health measurements. And that's the topic of this paper that's referenced here, that I think that a lot of our soil health metrics related to nitrogen, we might expect to work reasonably well in the low soil health system. They're not really meant for capturing the complexity of nitrogen available that we see in a healthy soil. And I'd also like to point out that when we build up these nitrogen pools and provide access to them, provide microbes and plants access to them, and we build up the carbon in the soil, that we're also creating a system that's more resilient. And on the left, you can see that system is totally dependent on nitrogen from fertilizer. There's very little recycled N. The plant available N pool is very, very large at any given time. It's very vulnerable to leaching, and you shut that valve off from the fertilizer, and that happens under a dry soil. That fertilizer N often becomes unavailable because it's getting to the plant via mass flow, and that system is very vulnerable to dry spells and potentially other sorts of disturbances that shut off that one pipe. Nitrogen fertilizer prices might be another factor. On the right, many nitrogen pathways increase nitrogen source resiliency. Nitrogen from fertilizers augmenting all of the internal sources of nitrogen. There's a lot of nitrogen recycling. It's a much tighter system, and it's a much more resilient system because there's multiple nitrogen sources, and if any one of them is shut off, the others continue to be available. And so carbon sequestration, I think, presents a real nitrogen dilemma. We're already facing a nitrogen crisis with regards to environmental quality and the loss of nitrogen. And if we're going to build up carbon in our ag soils to get them close or part of the way towards what our original prairies were, we're going to have to get a lot of nitrogen into those soils, and just saturating them in an organic nitrogen fertilizer is not going to do it. And I think as we move forward, we have to think about how we're managing management in terms of the use of legumes and cover crops to mop up nitrogen, and thereby build a more efficient system and rely on organic nitrogen inputs and sources. We have to balance internal and external N sources. That's another area that we've been working. What do different rates of external nitrogen applications do to internal nitrogen cycling? And we're trying to better understand that. We also have to continue to understand the mechanisms. I've been hearing for years from some circles that we know everything that we need to know about carbon and nitrogen, and now it's just a matter of getting the message across. I don't agree with that and think that there's so much work left to be done in terms of basic mechanisms. And it's also a very exciting time for monitoring and measuring nitrogen and developing new sensors and in situ ways to understand what's happening with nitrogen. And so maybe to be cheeky, we need to be replacing the 4 R's with the 3 M's and taking a holistic approach focused on overall management, mechanisms, and monitoring. And to close, I would just like to thank, this is just a very small number of the scientists who have come through my lab, but they're the ones whose papers I focused on today. They're amazing, they're inspiring. Every single one of these people is brilliant and doing amazing things, and I'm so fortunate to have worked with all of them. So thank you.
Well, thank you,
All
Stuart.
right.
You gave us a lot to think about there.
Yep.
And maybe to kind of thing that's come up the most is in the questions is this idea that it's not so much the pool of nitrogen or the rate of nitrogen cycling that we might measure. And, I don't think we have time to talk about, the new ways that we want to measure nitrogen, but it's more about changing the form of nitrogen in the soil. Would you say that's fair? That if you measure total nitrogen in the soil, we wouldn't expect it to be, we'd expect it to be going up with the management that you're talking about, but it's just these internal processes that are producing the nitrogen instead of having to rely on external sources. Is that?
Yes, I would tend to agree with that. I mean, I think that we still know very little about the processes by which well, at least from the standpoint of management and how we are going to more efficiently manage the pool, we don't know enough about the cycling of nitrogen through organic and inorganic forms and the role of the minerals, those mineral surfaces, the role of different minerals, the role of different microbes and their traits, the role of plant roots, exudation, mycorrhizae, and all of those complex interactions between them. And I think that with a deeper dive into those areas, that we have real potential to find some real important control points where we might be able to much more efficiently manage internal organic nitrogen cycling to create a system that looks a little more like our prairies. Of course, they're totally different. These are annual crops, root systems are different, but something that's somewhat more analogous in terms of having a much tighter nitrogen cycle where plants and microbes are able to generate inorganic N when they need it. And I don't want to sound, completely naive to the need for fertilizer as well. And so, what I imagine is augmenting internal processes and the buildup of organic nitrogen resources with external nitrogen fertilizer applications. There also are some agronomic considerations here as well. And what are our yield goals and what are we trying to achieve in terms of yield? And we have to carefully consider the relationship between yield and nitrogen availability and where we want to be on that curve, because it's a diminishing returns, especially as we get into the highest levels of yield and the highest levels of nitrogen fertility, where we see the most nitrogen losses. And so it really is a sort of a multi-pronged approach where we're thinking about the agronomy and yields and relationship to nitrogen, and we're thinking about external nitrogen inputs, and then there's my interests, which are really on the fundamental mechanisms and what are those plant mineral microbe interactions, and how do we better understand them, and how do we get to the point where we might be able to manage them to a degree and predict them in a model. Mm-hmm.
Great. Well, thanks, Stuart. We're a little past the hour now, so we're gonna have to let people go. But I want to thank both our speakers again for the excellent presentations. They both gave us a lot of insight and also a lot of questions to keep thinking about. So thank you again to our speakers, and we'll be taking a month off from the seminars, but we'll hopefully see you back here in July. So bye, everyone.